Hello, everyone. This is the start of a new chapter, chapter 9 on the cell cycle. We're going to be moving away from cell energies like we talked about in the enzyme chapter, cellular respiration, and the photosynthesis chapter, and move towards how cells divide. This is going to lead us into the next few chapters, which deal with heredity and genetics. But we got to get through how cells divide on their own in the body, and those are called body cells. This chapter centers around mitosis and how your body cells divide. The first part of this chapter is going to be looking about how DNA is structured within the nucleus. We all know that the majority, at least, of DNA is found in the nucleus for eukaryotic cells. So when we're looking at DNA, we're actually going to be looking at these things called chromosomes. Now, chromosomes are made out of DNA, but that DNA is packed tightly around histone proteins. These are just like packaging molecules, packaging proteins that the DNA uses to kind of assemble itself into a nice structure. So you can see the chromosome here. This is the full chromosome. These histones are little spheres, again, proteins that the DNA is going to wrap around, and they're going to wrap around until they make something called chromatin. Now, it's really important we know the differences between these vocab words. Chromatin is fiber made out of DNA and the histones. That's going to wrap itself around and tightly pack itself around. You can see all these loops here, and they're going to form chromosomes. The chromatids here are one individual arm. So here we can see that this chromosome has two chromatids. I know chromatin and chromatid have very similar and sounding words, and they're just literally one letter off. But understand this is a whole chromosome. We have one chromatin here, or sorry, chromatid here, and we have another chromatid here. Both of these chromatids are made out of chromatin. So I know this can be very confusing, but try to remember these three words: chromosome, chromatid, and chromatid. There's another way to look at the chromosome. Again, you can see the chromatin that's in the chromosome here. We're also going to have this place called the centromere, which is the middle of the chromosome where the two chromatids attach to each other. One misconception I want to start off by kind of just tackling is that the cell's DNA is usually in the form of a chromosome. That is incorrect. The majority of a cell's life, the chromosomes are going to be unpacked. And you can see this chromatin here. They will be in the chromatin form, but not the chromosome form. I always describe this as it's like a bowl of spaghetti. The spaghetti is not tightly packed together to form a chromosome. It's out and it's unpacked here in the nucleus. So majority of a cell's life, it's going to look like spaghetti, not like this chromosome form. The reason being is because we have to read this DNA. If it's it's packed tightly in the chromosome form here, you can't read it. It's like a closed book. You have to open up the DNA so our cell can read the DNA and read the instructions for life. Again, we're coming back to this chromatids. Each one of these is a chromatid. You can see here where the centromere is as well. One thing we're going to talk about is homologous chromosomes. Within homologous chromosomes, you have sister chromatids. This happens after replication. Replication is just the copying of the DNA in order to make enough DNA for the new cell to have enough DNA. So what's going to happen here is you're going to just copy and paste one of these chromatids to make two chromatids. You can see here, this cell here, or this chromosome here, has two chromatids. This chromosome here has two chromatids. This chromosome only has one chromatid. So a chromosome can be either one chromatid or two chromatids. This chromosome has one chromatid. This chromosome has one chromatid. This chromosome has two chromatids. This chromosome has two chromatids. These are sister chromatids. That means they're going to have identical DNA between the two. Like I said before, the centromere is just that location in the middle of the chromosome where the two sister chromatids are going to attach. You never will have non-sister chromatids attached to the centromere. It's only sister chromatids. The kinetochore is the actual reason that these spindle fibers, or actual region, I should say, that the spindle fibers are going to attach. We're going to see that later on when we talk about mitosis. So we'll come back to the kinetochores here. But just understand these two are the middle of the chromosome where the division is going to take place. All right, so here comes the confusing stuff. So the difference between diploid and haploid, we have to know this difference, and it's really difficult at first. I'll give you the easy part. Haploid cells have half of the chromosomes. So again, you got 23 chromosomes from your mom, 23 from your dad. Those came in the form of a sperm and an egg cell. they have half the genetic DNA. That means they have half the number of chromosomes. So you have 46 chromosomes in each one of your cells. The sperm and egg cell that made you had 23 of those chromosomes. A diploid cell, by the way, haploid cells, the only two haploid cells are sperm and egg. Those are the only two haploid cells. Diploid cells, they have the average number of chromosomes in the somatic cell. So for us, it's 46 chromosomes. And they have 2 chromosomes per pair. So we have 23 pairs of chromosomes in our cells. That's the diploid number. You can see here where there's 2 sets of chromosomes, or I should say 2 chromosomes per pairing for each pair of chromosomes in a diploid cell. You can see here there are 3 pairs of homologous chromosomes. So these guys are homologous chromosomes, these guys are homologous, and these guys are homologous chromosomes. One more thing I wanted to add in here is the concept of our homologous chromosomes. These again are pairs of chromosomes that have identical features like identical genes and the location of the centromere. These guys are homologous chromosomes. These guys are homologous. These guys are homologous. The blue and the green one are not homologous chromosomes here. The pink and the blue one are not homologous chromosomes. The only homologous chromosomes are the pink ones, the blue ones, and the green ones. Think of this as getting one from mom and one from dad, one from mom, one from dad, one from mom, one from dad.